Good afternoon, this is Dr. CB PhD, and today I'm going to be reading out Chapter 10 of Microbiology. This is a recording of 10:11:09. So let me begin. Now that we have introduced the main features of the molecular biology of cells and viruses, we can turn to the discussion of specific aspects of microbial genetics. In this chapter, we present the basic principles and techniques of bacteria genetics. First, we will discuss mutation and genetic recombination, and then we will explain how genetic material can be transferred from one organism to another. The transfer of genetic material from one organism to another can occur in a number of different ways in a microorganism, and if it is accompanied by genetic recombination, it can lead to profound changes in the organism. Finally, we will discuss some of the basic techniques in vitro DNA manipulation that have revolutionized the study of genetics of all organisms from bacteria to humans. Mutation and recombination. Mutation is an inherited change in the base sequence of a nucleic acid comprising of genome of an organism. Genetic recombination is a process by which genetic elements continue to separate genomes are brought together in one unit. Though this mechanism, new combinations of genes can arise even in the absence of mutations. Since the genetic elements brought together may enable the organism to carry out some new function, genetic recombination can result in adaptation to changing environments, whereas mutation usually brings about only a very small amount of genetic change in a cell. Genetic recombination usually involves much larger changes. Entire genes, sets genes, or even whole chromosomes are transferred between organisms. Prokaryotes, unlike many eukaryotes, do not reproduce sexually. However, there are mechanisms of genetic exchange in prokaryotes that, although considerably different from those involved in eukaryotic sexual reproduction, allow for both gene transfer and recombination. To detect genetic exchange between two organisms, it is necessary to employ genetic markers whose transfer can be detected. Genetically altered strains are used for this purpose, the alteration being due to one or more mutations in the DNA of the organism. These mutations may involve changes in only one or a few base pairs, or even the insertion of deletion of entire genomes. We begin this chapter on microbial genetics with a consideration of molecular mechanism of mutation and of the properties of mutant microorganisms as a prelude to our discussion of genetic exchange. Mutations and mutants. As previously mentioned, a mutant is a heritable change in the base sequence of a nucleic acid genome of an organism. In all cells, this nucleic acid is double-stranded DNA. A strain carrying such a change is called a mutant. A mutant, by definition, differs from its parental strain and genotype, a precise description of the genes an organism has, but in addition, the observable properties of the mutant. Its phenotype may also be altered relative to the parental phenotype. Parental strain. One would refer to this altered phenotype as a mutant phenotype. It is common to refer to a strain isolated from nature as a wild-type strain. Mutant derivatives can be obtained either from wild-type strains or from a strain derived from wild-type, for example, another mutant. Designating genotype and phenotype. Depending on the mutation, a mutant may or may not have a phenotype different from that of its parent. By convention in bacteria genetics, the genotype of an organism is designated by 3 lowercase letters followed by a capital letter, all in italics, indicating the particular gene involved. For example, the His-C gene in Escherichia coli codes for a protein that could be called the His-C protein. In this chapter, this protein, an enzyme in a biosynthetic pathway or histidine, is usually referred to by the name histidinal phosphate. Aminotransferase, which describes its enzymatic activity. However, some proteins, such as Rec A protein, do not have other names because enzyme functions can be difficult to describe in a few words. Mutations in the His-C gene would be designated as His-C1 and His-C2, and so on, the numbers referring to the order of isolation of the mutant strains. A phenotype of an organism is usually designated by a capital letter followed by two lowercase letters, with either a plus or a minus superscript to indicate the presence or absence of that property. For example, the His strain E. coli is capable of making its own histidine, whereas a His minus strain is not. Mutations in the His-C gene may lead to His phenotype if they eliminate the function of the gene product. Virtually any characteristic of an organism can be changed by mutation. However, some mutations are selectable, conferring some type of advantage on organisms possessing them, while others are non-selectable, even though they may lead to a very clear change in the phenotype of an organism. An example of such non-selectable mutation is that of loss of color in a pigmented organism. Such colonies usually have neither an advantage nor disadvantage over the pigmented parent colonies when grown on agar plates. There may be a selective advantage for pigmented organisms in nature. However, we can detect such mutations only by examining large numbers of colonies and looking for the different ones, a process called screening. We can detect such mutations only by examining large number of colonies. Note that the apparent color of a colony need not be because of pigment production. Mutant colonies of Halobacterium lacking gas vessels transmit light differentially than wild-type colonies and appear strikingly different on plates. Non-selectable mutants must be found by screening a large population of organisms, and the mutant phenotype may not be as easily recognized as the difference between pigmented and non-pigmented colonies. A selectable mutation, on the other hand, confers on the mutant an advantage under certain environmental conditions. So, the progeny of the mutant cells are able to outgrow and replace the parent. An example of a selectable mutation is drug resistance. An antibiotic-resistant mutant can grow in the presence of antibiotic concentrations that inhibit or kill the parent. However, the antibiotic-sensitive phenotype cannot be directly selected for by eliminating the antibiotic from the medium. It is relatively easy to detect and isolate selectable mutants by choosing the appropriate environmental conditions. Therefore, selection is an extremely powerful genetic tool, allowing the isolation of a single mutant from a population containing millions or even billions of parental organisms. Although screening is always more tedious than selection, for certain types of mutations, methods are available for screening large numbers of colors. For instance, nutritional mutants can be detected by the technique of replica plating. With the use of sterile velveteen cloth or filter paper on the imprint of colonies from master plate is made onto an agar plate lacking the nutrient. The colonies of the parental type will grow normally, whereas those of the mutant will not. Thus, the inability of colony to grow on the replica plate is a signal that it is a mutant, the colony on the master plate corresponding to the vacant spot on the replica plate can then be picked, purified, and characterized. A nutritional mutant that has a requirement for a growth factor is often called the auxotroph and the wild type. Parent from which auxotroph was derived is called a prototroph. For instance, mutants of Escherichia coli with His- phenotype are said to be histidine auxotrophs. Although of great utility, replica plating is a screening process, and it can be laborious, is isolated from mutants by screening. Penicillin selection. An igneous method widely used to isolate auxotrophs is the penicillin selection method. Ordinarily, mutants that require growth factors are at a disadvantage in competition with the parent cells. and so there is no direct way of isolating them. However, penicillin kills only growing cells, and if penicillin is added to a population growing in a medium lacking the growth factor required by the derived mutant, the parent cells will be killed, whereas the non-growing mutant cells will be unaffected. Thus, after preliminary incubation in the absence of growth factor in a penicillin-containing medium, the population is washed free of penicillin and transferred to plates containing the growth factor. Among the colonies that grow up, including some wild-type cells that have escaped penicillin killing, should be some growth factor mutants. Penicillin selection is a key kind of negative selection. The selection is not for a mutant but against the parental type. Some of the most common kinds of mutants and the means by which they are detected are listed. Mutations can also occur during replication as a result of errors in the pairing of bases, leading to changes in replicated DNA. Mutations involve one, or a very few, base pairs are sometimes referred to as point mutations. Point mutations can result in base pair substitutions in DNA and the insertion of deletion of a base pair called microinsertions and microdeletions. As is the case with all mutations, the phenotypic change that comes about because of a point mutation depends on exactly where the mutations took place in the gene, what the nucleotide change was, and what product the gene normally encodes. Base pair substitutions. If a point mutation occurs within the coding region of a gene that encodes a protein, any change in the phenotype of the cells is almost certainly the result of a change in the amino acid sequence of the protein being produced. Number of base pair substitutions that can occur in a short region of DNA with a gene that encodes a protein. The error in the DNA is transcribed in the mRNA, and the error in this mRNA, in turn, is used as a template and translated into protein. Because only one strand of DNA is used as a template for the mRNA, an AT base pair does not have the same meaning as a TA base pair. The triplet code that directs the insertion of the amino acid via transfer RNA will thus be incorrect. What are the consequences of base pair substitutions? In interpreting the results of mutation, we must first recall that the genetic code is degenerate. Because of the degeneracy, not all mutations in protein encoding genes result in changes in protein. This is illustrated, which shows several possible results when the DNA that encodes a signal tyrosine codon undergoes mutation. As seen, a change in the RNA from UAC to UAU would have no apparent effect because UAU is also a tyrosine codon. Mutations that give rise to such changes are called silent mutations. Note that silent mutations in coding regions almost always occur in the third base of the codon, arginine and leucine, can also have a silent mutation in the first position. As seen in Chapter 7, changes in the first or second base of the triplet much more often lead to significant changes in the protein. For instance, a single base change from UAC to AAC would result in a change in the protein from tyrosine to asparagine.